Welcome to Nefertari Audiogenetics. For decades, we thought that genetic code is fixed. Each codon specifies one amino acid and that's it. But emerging research shows that ribosomes can sometimes read the code in non-standard ways, producing protein variants that are not strictly encoded in the DNA. These variants is called alternative RNA decoding. In standard translation, messenger RNA is read 3 nucleotides at a time. Each codon matches to a specific amino acid by transfer RNA. But in alternative RNA decoding, this match is not always perfect. 3 mechanisms are behind this. The first is sense codon recoding. A codon normally coding for one amino acid is read as another without any mutation in the DNA or RNA. The second is codon-anticodon mismatches. The transfer RNA anticodon does not perfectly match the mRNA codon, yet translation proceeds inserting a different amino acid. Finally, context-dependent decoding. Local RNA sequence, RNA modifications, and transfer RNA availability bias which amino acid gets inserted. Recent proteomes-wide studies in human and mice identified tens of thousands of amino acid substitutions from alternative translation across nearly 2,000 genes. In hundreds of cases, the recoded proteins is even more abundant than the standard forms. Alternative decoding expands the functional proteome without changing the genome. It helps explain why protein levels often do not match the messenger RNA levels, how cells rapidly fine-tune responses to stress or disease without waiting for transcription. But what allows the ribosome to break the rules? Transfer RNA modifications, especially at the flexible third codon position, reshape codon-anticodon pairing and can promote near cognate reading. RNA modifications on messenger RNA alter local structure and decoding dynamics. Finally, coding usage and transfer RNA pool differs between tissues, biasing which codon are decoded efficiently and how accurately. Some recoded variants are more stable, so they accumulate to higher levels than standard proteins. Alternative decoding can be used in many fields, such as biomarker discovery. A tissue-specific and cancer-specific recoded proteins are being explored as new diagnostic markers. Or in precision oncology, proteoforms data can refine tumor classification beyond genomics and transcriptomics. Or neurodegeneration research. Recoded proteins linked to misfolding are opening new angles in Alzheimer's and Parkinson's research. So, in animals, alternative decoding is increasingly recognized as a major source of proteome diversity with clear disease links. However, in plant, diversification relies more on alternative splicing and rapid transcriptional reprogramming under stress with less proteome levels evidence for widespread sense codon recoding so far. So, genetic coding is not as rigid as we once thought it is. Translation itself is a hidden layer of regulation, one that allows cells to generate new protein variants in real time. Thank you for listening to Nefertari Audiogenetics.